today we are going to study about museum techniques. Now, what are museum techniques? Museum techniques are some techniques which used to preserve the specimen for pathological museums or pathological laboratories which come for learning purposes. So this is main question in which in third year exam. So there are 8 steps of museum technique. First is reception, second is preparation, third is fixation, fourth color restoration, fifth preservation, mounting, special methods and presentation. Reception. So what is reception? The specimens are mostly collected from the teaching hospitals. Surgically restricted specimens from operation theatres, necroscopy, specimen from postmortem group, research laboratory, specimen then should be received with full details of the patient. Then preparation of the specimen. Contact of the specimen with tap water is prohibited. But what happens if we contact this with tap water, then necrosis may take place. So it should be preserved in what? Normal saline or formalin. Normal saline is fine. So in normal saline it should be washed. Okay. Then drying greases the surface. Therefore it should be kept wet in normal saline only. Should not be kept in saline for more than 2 hours as autolysis sets in. So it should be fixed, right? Now fixation. Objective: to preserve the cells and tissue constituents in as close to life state as possible. Now fixation stops autolysis, bacteria decomposition and stabilization the cellular and tissue. Fixative used are based on formalin fixative techniques from Kaiserling technique. Now these are Kaiserling techniques we will study later. Kaiserling recommended that initial fixation should be in neutral formalin, that is Ki solution, and then Kaiserling solution, preserving in glycerin solution, that is Ki3, Ki1 and Ki3. These solutions are preserve color. Now fixation and color restoration. Now color of the specimen, which gives the idea about the disease or the normalcy of that this. So color should be also preserved. Then next is preservation. Yes, and mounting. Mounting, now we see big jars in the laboratories of pathology where liver, lungs, brain are preserved. Okay. Then special methods and presentation. So this is the basic of museum techniques which I told you today. This I will take in detail. So thank you for joining me today, and let's plan something else. So please ask me questions about some other things which you want as a topic. So thank you. So how is method? Do you like it? Okay. Bye bye.